I'm a second year PhD student. I work in the nutrition department, and my research area is focused into nutritional biochemistry. And my research is on basically looking at the biological pathways of how omega-3 polyunsaturated fatty acids, such as EPA, DHA, can help with resolving inflammation, particularly pulmonary inflammation. And I use different mouse models to test that. The method I use is a common method that you might have heard of is ELISAs, which stands for enzyme-linked immunosorbent assay. So basically, for an ELISA, you have a 96-well plate. The plate is coated with an antibody, which is basically a marker that can attach to the protein that you want to measure. So for my study, I measure proteins such as cytokines, and those are chemical messengers that are released from immune cells. So I want to see how much are the immune cells responding to the disease. So I coat my plate, then I put in my sample. And then that, after a couple of other steps, taking up about 5 hours, I get a range of colors. This is another plate showing some range of colors. And the darker the color is, the more protein there is. Then I put the plate into this plate reader. And for example, this is where my plate would go. And then it will generate a spectrum that will measure at a particular wavelength. And it will draw out a standard curve with my standards that tell you what concentrations are. And those concentrations, if you know the unknowns, you can extrapolate what your unknown concentration is for your protein. I then use a software called GraphPad to analyze my data. And I put in my data into GraphPad, and that generates another standard curve. And once I extrapolate a protein using statistical tests, I create a table and graph to look at the differences between my control and experimental groups. I can also use the software to generate figures for publication. My favorite thing is going to conferences and collaborating and networking with other scientists, because they always give me a new perspective to look at for any given scientific problem. And that really challenges my critical thinking, so I really enjoy that. My advice is make sure research is for you. So please start volunteering in research labs, either in your university or a university near you. You can also get paid if you work as a work study in a lab. We actually have a couple of work studies, and they've actually gotten published in our papers as well. So it's a great start. There's also many summer research opportunities. So look early, and you will be able to see if this is what you want to do.